Very excited for the conversation that we're going to be having with him. So without any further ado, let's bring him on to the show. Please help me welcome Jonathan.
Hey.
Oh, thank you for coming.
Beautiful. What an intro. Really great. Great to be
better
here.
at it.
Right on. Well, first of all, congratulations on the launch. Big accomplishment. Is this your first time launching a company?
Yes, it is. It is. And the last few days have been a bit of a whirlwind. We've had companies reach out to us. We've had people from kind of all over the ecosystem basically say, thank God you exist, which is great, which is a nice proof point for us. And it's been really great to see. It's really exciting and looking forward to telling you guys more about it.
Excellent.
Yeah, me and Ben have a lot of not a lot of experience, but me and Ben have a lot of knowledge around the biology of this, like the DNA recognition pathways and things like that, and how the cells get pretty pissed off. They get tricked into thinking that they're actually virally infected when you're trying to edit them, essentially. And it turns out to be a pretty big problem in both like DNA-based therapeutics, but mainly in things like CAR T cell generation or any kind of like cell manipulation. So tell us a little bit about what you're doing and where the idea came from. That's kind of where I want to.
If it's okay, let's just back up a little bit for the uninitiated. What is the problem here? Just in case people are coming in a little bit more fresh, why should we have to worry at all about stealthing DNA inside of cells when we're trying to therapeutically manipulate them?
Yeah, so I think you guys have nailed it at the technical level, which is the things the DNA, the therapeutic DNA that we put into patients and into cells looks nothing like our endogenous genes. And as such, it triggers a lot of antiviral pathways, which luckily defend our cells from things like HIV or viruses or other mobile elements. But essentially, it shuts it off. And what this leads to is a couple of things. So firstly, in the context of gene therapies, we see sometimes during clinical trial timeframes or sometimes post-launch that you get an initial efficacy, which can be stunning, but it can fade over time. And that leads to disease relapse. These things often can only be dosed once. And then an interconnected problem is that because most of the DNA you're putting into a patient is turned off, we have to dose patients with insane amounts of drug. So we're giving like 1,000 trillion DNA copies to a patient. And just to produce that much material costs about $1 million a dose. Yeah. So then you have these two interconnected problems. You have enormous cost, which might work if that therapy did last for the lifetime of that patient, right? The healthcare economics might work. But if it doesn't, the whole kind of economic underpinnings falls apart. And actually, that's what we're seeing. So we're seeing a kind of market adoption crisis for gene therapies. We do have amazing proof points in Luxturna, Zolgensma, others that are making people see for the first time or enabling kids to walk again. The power, the kind of proof point is there. But for gene therapies at large, this kind of commercial bottleneck is just creating this chasm between the therapeutic and commercial potential of these therapies and then the patients, So that's the problem that we're really trying to solve.
Very good. Yes. Well stated. So folks, if you have any questions for Jonathan, I'm sure he'll take them in the comments. Just drop them down below and we'll get to those. So yeah, I really appreciate that overview of the problem here. So the question now becomes, like why have other approaches failed? Like what have people tried to do in the past to kind of get around this issue and it hasn't really worked?
I think it really comes down to the fact that we've been shooting in the dark for a long time. So we have a few kind of parts of a toolkit, things like introns or WPRE or codon optimization or other kind of strategies that have been around to improve the efficiency and expression of DNA. But we haven't really had a mechanistic understanding of why DNA is silenced and what's going on there. And I'm kind of curious for you guys, interested to see whether you've heard much about the Hush complex, because Hush was discovered in 2015, so just about 6 years ago. And I was lucky enough to do my PhD with Professor Lehner at Cambridge, who discovered Hush. And initially, it was kind of an anti-HIV factor. And then this really cool thing happened when we realized it wasn't just HIV that Hush was turning off. Really, Hush was finding therapeutic DNA, any kind of exogenous DNA, and massively shutting it down. So once we then had that mechanistic understanding, I spent my PhD really working out what is the self and non-self signal on the molecular level, and how can we use that insight to design DNA that can evade the Hush complex and therefore not be silenced inside cells. So I think that's really our kind of unfair advantage of what we're doing at Houdini.
Oh, you discovered it? That's the unfair advantage. That's quite the advantage. Yeah. So my experience is mainly in the immunological consequences of like viral infection, and how like DNA damage can lead to micronuclei formation and like cytosolic DNA and CGAS and sting activation and like interferons and all kinds of stuff like that. I don't know what the Hush complex is, but it's very interesting to know that we have a intracellular nucleic acid analog to like self and non-self, such that we do at the peptide level on the extracellular surface. So what is the Hush complex? Is it analogous to like the self and non-self, but maybe more ancient?
Yeah, that's it really. And it's incredible when I speak to people in the field, and really nobody has heard of Hush yet. And I think that gives me a lot of hope, because I think by kind of having this unlock and knowing how it works, we are able to target it in a much more kind of effective way. In terms of what Hush is, it senses transcript. So it's really a first pass kind of genomic defense system. So it's really interested in anything that ends up in the genome and is active. So if it's not active, it doesn't care, because it doesn't need to. If it is active, it's evolved to sense that transcriptional activity, and then it essentially remodels chromatin around that locus or around that piece of DNA to just shut down transcription and shut down activity. So probably some ancestor of the Ritz complex or other things you see in the East. Really, really cool.
So does it have to do with recognition of various like viral promoters and stuff, or how does it actually? Or I guess obviously some viruses can replicate. How do they get around it? Do you guys use some kind of similar thing to like maybe evade, or are you trying to emulate self, or is it the same thing?
It can be the same thing. So there's different ways that certain elements can do it. Sometimes viruses don't want to get around Hush. So for example, in the case of HIV, although it does produce proteins that take Hush out, it can go latent by kind of exploiting that silencing. And that's responsible for this latent HIV and memory T cells. So yes, to your question, we can learn a lot from virology in terms of Hush escape mechanisms and how it's been done. And yeah, we're definitely inspired a lot by those types of things.
Excellent. Okay, there is just one question here in the chat, which I think I'm just piecing together the different pieces here. So whereabouts did you study in this? And this was part of your graduate work, correct?
Yeah, so this was at Cambridge, and it was during my PhD.
Right.
So 2019 to 2023. Right. And then it should be said, for me, I knew I wanted to kind of turn this into a company. I didn't even know what VC was at the time. So I was like, dude, I need to figure out how to actually build a company. And I was able to get a job at Deep Science Ventures in London for a couple of years. And Kirsten there was my former boss, and kind of taught me the tricks of the trade, and then encouraged me when the time was right to return to that research, introduced me to my better half, which is a guy called Lee Dunham, who's the co-founder, and was working on the commercial side as BD director at Catapult, cell and gene therapy Catapult. And yeah, we basically got together. We were super excited, and we quit our jobs about a year ago and started building this out.
Nice.
So Jonathan, I graduated with my PhD in 2022 and did a couple of years of a postdoc. So I'm still in the academic system. I get out of the academic system and I'm doing like scientific communications. And one of my friends on LinkedIn had gotten a new job at this company called RA Capital. Okay. I'd had no idea what venture capital was either. So I messaged him and I was like, oh my gosh, awesome. Like, can I come work for your new venture capital company? And he was like, bruh, RA Capital is like the biggest venture capital company in like biotech. I had no idea. The academic system does not do a very good job at like teaching scientists how to become entrepreneurs. It's like so bad at it. Maybe we can like have an initiative, Ben, where we kind of help penetrate that market, because you come out of it and it's just like, wait a minute, I can make money with my therapeutic. I can actually, or even like before that, like in your first year of your PhD, like you should be conducting experiments with the goal of like identifying therapeutic candidates and like doing IND enabling studies and like things like that in your PhD, but you just don't, right? So don't feel bad about being ignorant, because I feel like most people are. Right.
I can't let that go by without plugging something that Deep Science Ventures started. So it's called the Venture Science Doctorate. And actually, it's really exactly what you said. So it's a PhD program, which is really built around, you know, the final output of the PhD should be a company.
Yeah, 100%.
Heck yes.
Creation as part of that PhD program. So that's a plug, but an amazing initiative. I think there should be more things like that.
Isaiah, clip that. There you go. Okay. Yeah, that's super cool. And I think especially in like the academic climate right now, with like funding is going dry and like, you know, you probably know the experience. I was a postdoc trying to get an associate professorship role, and it's just like, good luck. You're going to be in the middle of Oklahoma at some like junior college, right? It's like, there's no jobs around. So where do you go? It's like, well, you go into industry, but most people are just like, well, let's go and be a pipette machine and at some, you know, biotech company. It's like, well, you could have started your own company if you started this a little bit earlier.
100%.
So, yeah.
All right. So just to come around a little bit here. So you've identified this complex. Other approaches kind of have focused on like other just unknown, like known pathways, right? So I used to work for Virka Biotech, and they're still going strong up in Ottawa, right? They have small molecules that transiently attenuate some of these different antiviral defense pathways. And you know, they've seen some pretty interesting results, both on the therapeutic side and on the production of virus, like as well, for like AAV and lentivirus and that kind of thing. So just to kind of like separate the two of those, what is the approach here? Are you guys like developing a small molecule that helps activate this complex, or are you how are you modulating this complex to help out with therapeutic delivery of DNA?
Yeah, it's a great question. So I think the key distinction here is that we are encoding resistance to silencing, resistance to hush within the DNA itself.
The DNA.
So we design the DNA according to proprietary design rules. We have an AI model that plugs into our kind of wet lab closed loop system, and we can redesign a whole transgene. But we also have these things called Houdini elements. So these are like really, really small. Sometimes
is one of them called the straitjacket?
Not yet, but maybe the next one. We're finding new ones all the time, and they're these really small, kind of 100 base pair sequences that we can just plug and play into somebody's therapy, and it basically is a self-hallmark. So you're essentially giving it this kind of camouflage against the hush complex, and the reason we do that at the level of DNA is because, particularly in the gene therapy context, you really don't want to be messing around with these systems. It very well could kill somebody. I mean, these are essential genes in all of our cells in the body. So we need to encode resistance within the DNA itself rather than globally mess around with the pathway. Things like endogenous retroviruses and transposons and other things which would be jumping around. And I think, that's also the case I mean, I'd be interested maybe offline in kind of how the kind of global approach works in biomanufacturing, because something, we do some work in the context of biomanufacturing, so in CHO cells, and we're looking at complex antibody formats and trying to increase the yield there. But again, we think that CMC wise, much better to do this at the level of the DNA rather than globally start reactivating a lot of potentially dangerous stuff.
Yeah, you couldn't give. I think that a lot of it is just like antiviral machinery inhibitors, like sting inhibitors and things like that. And it's like you couldn't do that systemically. You'd have reactivation of all kinds of stuff, and it wouldn't be good.
Exactly.
So
exactly.
And unpredictable
I'd say
too.
that was the kind of key distinction there.
Yeah, and it sounds like this is it's a much more elegant approach. That's like, no, we're going to mask our therapeutic, leaving everything else intact.
Exactly.
Yeah.
We tried pretty hard. I didn't quite get there by the end of my PhD, but I'd done enough that by the time we were able to use some kind of neural network models and machine learning models on top of the stuff that we knew, that really made the difference for us. And then we were able to find things that you just couldn't find with kind of like manual searching or simple bioinformatics. So yeah.
So I'm curious, I don't want to spend too much time on this, and maybe it's proprietary, I don't know, but was the training data for that like publicly available data, like things like viral genomes and things, or was it your own internal proprietary data sets that you had created?
Yeah, it was all internal.
Okay, that makes sense.
For us. I think it's really important for us just to, so we have libraries of different Houdini elements and versions of transgenes and so on. And we're just building that up as an internal resource so that we can train those models, keep it internal, close that loop. And we're already starting to see the model kind of prove out with second gen versions that are better than the first. So we think we're on the right path there moving forward.
Excellent. Love it. Okay, folks, if you have any questions for Jonathan, feel free to keep them coming. There is another question just here in the chat, which is what was your PhD in?
So it was really, it started as an HIV project, thinking about the Hush complex, and then it turned into figuring out what Hush likes and doesn't like, essentially. So it was really all about the molecular biology of the Hush complex.
Nice.
It's funny how those kind of things work out. Like, who would have thought that you'd be here starting with HIV? That's crazy.
Mental.
Yeah. So are you guys planning on doing it mainly for DNA-based therapeutics, or is it mainly for like ex vivo cell engineering and things like that, or both?
Yeah, I think one of the problems, like it's a good problem to have, but we could go in so many different ways. And so as part of the work that we've been doing over the last 6, 7, 8 months, we've kind of gone quite wide. But what we're really looking to do is focus. That probably means a little bit more emphasis on the gene therapy side of things rather than the biomanufacturing. But that does include things like kind of CAR-T. That could be in vivo, but it could be ex vivo. Something that's really interesting is from clinical studies that the expression of the CAR receptor also fades with time. And you can see that the DNA is still there, but the transcript and the kind of protein drops off. And you can actually map that back to relapse in patients that get CAR-T therapy. So we think that we could potentially be trying to prolong the expression of CAR receptors in vivo or ex vivo to try and prevent that relapse. So it's something we're looking at as well. We've got a few angles on the table.
Right on. Okay, so I guess the follow-up question here, right, is okay. So you're doing all this work on the Hush complex. Like, what was kind of the initial pieces of data where you're like, "Holy moly, we've got something here"? What was kind of that aha moment?
I think it was probably a little bit less sexy than that, and a little bit more.
It always is. It's always very mundane and
it's
like
a slow burn over
a random
Tuesday.
This thing happened.
It was a slow burn. At the beginning, all of us thought there's a good chance this will work. There was a part of me that was like, will this work? And really, most things we tried worked to some degree or another. And by work, I mean, we benchmark expression per dose against industry standards, state of the art for different payloads. I mean, we've tested upwards of 10 different therapies. We've tested AAVs, lentis, normal DNA, transposon delivery systems, different cell types, and everything works to a degree. Something that I found really satisfying recently is we were able to take an FDA-approved construct, Roctavian, which unfortunately was pulled from the market. So it's a factor VIII therapy, pulled from the market because of the silencing. You saw that there was a drop in efficacy over time. It cost so much. We thought, let's see if we can improve it. And at least in the dish, in liver cells, we were able to kind of 4 or 5x the per dose expression of that. And so to see that work using a kind of FDA approved benchmark was pretty awesome.
So you could resurrect a therapy. That's
I don't know if that's going to be our model. I don't know if that should be our model, but absolutely, definitely working in collaboration with partner companies. We'd be super excited to kind of work on some of their assets that could be realistically it would be pre-IND stage. But what we'd really love to do is work with pharma companies across different asset pipelines, and just make sure that we can integrate early so that these gene therapy pipelines aren't just stalling at the, as soon as you get to a trial or even post-launch, and then you end up having to pull it from the market. So that's really the kind of end goal for us.
But I would imagine that big pharma probably has like shelves on shelves of discontinued DNA-based gene therapies. Like, have you guys?
Jonathan knows.
I'm gonna say,
probably
like,
got some very interesting chats over the weekend.
Are we gonna see like a Merck Houdini Bio licensing agreement in 3 months for $45 billion?
I think the opportunity, you're on the money in terms of the opportunity there, but it's also about future pipelines, right? It's not just about things that haven't worked so far. It's really about looking ahead. And gene therapies are getting more complex. They're getting less self-like, more synthetic.
Yeah, true. Good point. Yeah.
And we think that they're going to be more and more prone to hush silencing and kind of shut down inside the cell. So we're also thinking about the value that this could add to future pipelines for pharma, as well as thinking about the current ones.
And does this affect any kind of, I'm not an AAV guy, but does it affect, do these Houdini elements affect like AAV packaging or whatever the right term would be? Is it completely inert to that? We've
done some initial CMC, which is really positive in terms of the impact of that in packaging. So we're able to see full length AAV virus package within the capsid. We need to do more. We've been up and running in the lab properly as a company for about 7 or 8 months. So we've been
like 2 days.
Yeah, we've been moving super fast. And it's something that, we're really focused on. We're working with a couple of partners as well, with better analytics capabilities to like really dive into that, because it's an important question as we go forward for sure.
Nice. Well, Jonathan, like just like a separate idea here, like it might actually make sense. I don't know if you guys have had any chats with my old company, Virica Biotech, but they're very good at doing high throughput screens and stuff like that. So might be some sort of potential 1 + 1 = 3 situation there. No, I'm no longer affiliated with them, but good
Ben's getting paid on commission
over there. Part of the Commonwealth. Get out of here, Jeff, with your Independence Day.
Yeah.
Let's do it. Let's have the discussion.
Nice. All right.
But I feel like anybody doing a DNA-based genetic medicine needs to know about this. Like, why wouldn't you incorporate it?
Yeah, that's it. And to be honest, the headwind that we have as a company, I think, is really fueled by the depth of the problem. So really, the massive majority of therapy developers that we're engaging with, the first thing they say is like, yes, this is a problem for us. And it's really, the way I see it is, I feel like there's a risk of throwing the baby out with the bathwater for gene therapies. And like I said, right at the start, it's literally curing people of disease that we could never treat before. And really, I think this is the key bottleneck, and it's a tractable one. It's we know how it works. We're finding ways around it. If we really are able to crack it, it could completely flip the economics, the therapeutic efficacy, and that would be incredible. That's kind of the massive, holy grail at the end of it, is that actually more patients are able to access gene therapies, and also they can make money, right? There's so much value in that as well. So that would be my message. Don't give up on gene therapies.
Excellent. Love it. Well, I know we've only got a couple minutes left here. We want to be super respectful of your time. So we did just get another question in the comments here. Like who's backing you? Who are the investors for this initial round?
Yeah, so we've got an awesome little syndicate of investors. So Science Creates VC are our lead investors on the pre-seed, and Harry and the team there are awesome. I don't know if you know any of them. They're based down in Bristol. Harry exited Xylo Therapeutics. It was a Bristol spin out, about 800 million, and then set up SCVC.
Nice work.
And he's kind of matches, he matches our intensity, which is great. Nice. And then there's also Kirsten, who's incredible, and that's Deep Science Ventures that are also part of our investors. And then Cambridge Enterprise VC as well. So a few at the moment, and we're really looking ahead towards our seed raise now. So we're really trying to build up some momentum there and get some more capital into double down.
Excellent. Yeah. So I guess on that note, right? Like what's next over the coming months? What's going to be obviously, raising more money, but what will be the key data that you'd like to generate ahead of that next raise?
Yeah, really for us, it's about moving into more in vivo models, disease models, not just any disease models, but gold standard models for the indications that we're working on, and really proving out our technology more in that in vivo setting, reading out on therapeutic efficacy. We're also really keen on diving deeper into CMC and really understanding what that looks like. So far, really, really encouraging, but we're looking to flesh out that data package. And then on the kind of commercial side, it's really about trying to find some pharma partners that would be interested in working with us across their assets, and then being able to kind of prove out and validate the technology in a real world live example.
Nice. I love it. So I'll be honest. I very much underestimated the potential of this technology when I first heard about it. I was like, oh, it'll be good for CAR T cells, whatever. This could actually be implemented across the entire universe of genetic cell therapy or gene therapy. Sorry. It's actually good for you. Congratulations. That's amazing. And especially being founded on the key inventor / discoverer, your moat is just so deep and wide. That's incredible.
Really appreciate that. We're just as excited. It's kind of quite surreal. But we're really going for it, and we totally agree that there is the potential here, if it works as well as we think it will, to make a massive impact. So we're super stoked.
Excellent. Well, Jonathan, again, thank you so much for your time. We really appreciate you coming on. We were fans before. We're even bigger fans now. We're really looking forward to seeing you guys fly in the coming months. We look forward to the big announcement with Merck or someone in 4 months from now, and you get you get bought for $700 billion.
Beautiful. Thank you so much for having me on, and let's chat offline as well. Really keen to kind of continue those conversations as well.
Yeah, fantastic. Sure. Excellent. Thanks so much again, Jonathan. Cheers.
Good meeting you, man.
Cheers. If you like this video, subscribe for more. We've got a boatload of content coming. Also, Who should we talk to next? Drop your suggestions down below.